Warm welcome to everyone to this webinar, and thank you all for being here. My name is Farnoosh Safavi. I'm the head of education department, and I'll be gladly hosting today's webinar, which is on precise motor mapping with TMS. Today's session will be presented by Dr. Konstantin Weise, who studied theoretical electrical engineering at the Technische Universität Ilmenau and received his PhD in 2016. From 2016 to 2022, he was working as a postdoc at the Max-Planck Institute for Human, Cognitive and Brain Sciences in Leipzig on methodological advancements and numerical modeling of TMS. From 2022 to 2023, he was working in the Department of Clinical Medicine at Aarhus University in the field of Tumor Treating Fields. From 2023, he is a temporary professor for electrical engineering at the University of Applied Sciences in Leipzig and the principal investigator at the MPI for Human, Cognitive and Brain Sciences in Leipzig. After the scientific presentation, there will be a short practical demo by ANG Neuro application team. And we will be answering questions afterwards, like always. In the course of the presentation, you also have the option to enter your questions in the chat box. As you're by now familiar with the poll questions, you will receive some in the course of the webinar. And it will be highly appreciated if you take part in those poll questions to make it as interactive as possible. So, Konstantin, we very much appreciate you being here with us today and accepting our invitation. Without further ado, we are looking forward to your presentation. Thank you.
Yeah, thank you very much for this kind introduction. I'm also very happy to be here today and to present you our latest methodological advancements in TMS motor mapping. And this work was done at the MPI in Leipzig together with Ole Lumsen, Benjamin Kaloch, Axel Thielcher, Gesa and Thomas. And thank you very much for all of my collaborators here. So without further ado, I want to just start with my presentation. So what we are doing here. So transcranial magnetic stimulation, just a very short introduction. So we are putting a coil on top of the head, and we are running a large current through it with a frequency of 3 kilohertz. And we are only using one sine wave, so one period of a sine wave here. And the magnetic field which is generated by this high current is time changing and inducing an electric field in the brain. And this electric field in the brain excites the neurons, basically in our neocortex. And in consequence, if you put the TMS coil above the motor cortex, we are exciting the upper motor neurons, and their signals are traveling down the spinal cord until they reach the lower motor neurons, the alpha motor neurons in the spinal cord, and then the signals are again modulated and sent through peripheral nerve fibers to the muscles. And at the muscles, we have the opportunity to measure these incoming signals, and this is what we call the motor evoked potentials. And here you see an example of a motor evoked potential measured at the FDI muscles, the index finger. And what we are usually looking at is the peak-to-peak value. So the higher we are stimulating the cortex, the higher is also the muscle response and the peak-to-peak value of this motor evoked potential. So the question now is that we want to localize these neurons in the cortex which were actually stimulated, and this is a challenging task because the anatomy of our brain is so different for different people. So we cannot easily say from one person to the next, okay, your FDI representation or your hand muscle representation is there or there. So we have to repeat it for every person individually. So, and how can we do this? So first we are looking at a very traditional approach, how motor mapping was working for the last, I don't know, 25 years. And this classical mapping approach is based on a grid. So you define a grid on the surface of the skin surface of the head, and then you simply apply TMS and you record the peak-to-peak value of your motor evoked potential, and then you give every, let's say, grid point, you assign this peak-to-peak value. And here, for example, we would have the highest peak-to-peak value if the coil would be positioned here at the dark red spot, and then the simplest approach to localize, let's say, the neurons is to project down basically the to the cortex, so from the coil to the cortex. And this is obviously a very rough approximation. And for example, the true spot would lie here, for example, in our projection approach would result, for example, here in the premotor cortex, which is of course not true because of this rough approximation. So, and this brings me basically to my first poll question. If you do TMS mapping, yes or no, or are you experienced with it? Do you, yes, it's something. So let's wait for the results a little bit.
So already more than 70% voted, so I close the poll and share the results.
Okay, so we have the majority which is who are not doing TMS mapping, but still 20% are doing it regularly, which is also very nice. So maybe you find out something new here, what you could also use in your lab. So let's come on the way. So now we want to find a better way basically to do it. So the projection approach is obviously not the most accurate one and comes with a lot of assumptions. So, but what we did not consider yet is the TMS-induced electric field in the brain. So when you put the coil somewhere on the skin surface, we will induce an electric field, and the electric field distribution, of course, highly depends on the underlying geometry here. So, and here I show you some examples how the electric field changes when we simply move the coil around. So not only the distribution and the shape changes, also the magnitude changes by far. For example, here in the somatomotor cortex, we have a decrease, for example, or here, if you would apply the same stimulation strength in the dorsolateral prefrontal cortex, we also get a lower field, in a similar way also the inferior parietal lobe. So, and this induced electric field can be calculated using numerical tools like the finite element method or the boundary element method or any other numerical tool. And this now brings me to the basic idea of what we have here now in our TMS mapping problem. So on the one hand, we have the TMS effect. So we have the behavioral output, what we see, for example, the motor evoked potential. And this motor evoked potential depends on the coil position orientation and on the stimulation intensity. And on the other hand, we have the electric field, as we just saw in the previous slide. And now the question is, how can we combine both information? So now we want to make use of the electric field distribution in the brain in order to improve our mapping. So, and this comes to the next question. So now only 20% maybe can answer this question. If you do TMS mapping, how do you do it? So the first answer would be a grid-based approach using the projection technique. The second answer could be you're using a grid-based approach with electric field information. Or option number 3 is you use your own approach without the knowledge of the electric field, or you use your own approach with the knowledge of the electric field. So would be very interesting to know how basically you are doing it currently.
All right, since more than 20% voted, and that's the number that already has experience with TMS, I close the poll and let's see the results.
This is I didn't expect that. So we have 15% who are using the grid-based approach. Then we have 43% who are using grid-based approach with electric field information. You have 29% using an own approach without the electric field, and 14% an own approach with the electric field. So we have approximately a bit more than 50% are using electric field information, and the other, a bit less than half, is not using electric field information. So this is very interesting, and maybe also when I continue now, maybe I can convince the other 50% maybe to use this information. But this brings me directly to the next question. For the ones who are not using electric field information in TMS studies, do you think the first option would be you do not think it's improving the accuracy of TMS? The second option would be that it's currently too complicated and you are not interested. Or the third option is it's too complicated, but you would like to use it if it's easier to apply. And the last option is you are already using electric field simulations. So I'm expecting 50% in D now. Okay, 20% are not believing that it's improving the accuracy of TMS. 40% that it's currently too complicated, but they would like to use it, which is a very good result. And we have also 40% that are not using, that they are already using electric field simulations. So this is very interesting also for us, of course, to develop maybe scripts and tools which are easy to use in the future, and where it is easier to maybe combine the information from different neural navigation systems into your numerical simulations. So this is, of course, also for us very interesting. Okay, thank you. So we can continue here. So now I want to come to the actual mapping approach we developed. So it is in total we have here 5 stages we can go through. And I want to introduce now a step-by-step introduction here. We published a protocol which is describing each step in very good detail. And this is not in print, so this is it's already published in Nature Protocols. And maybe if you're interested in this protocol, you can have a look at it and go step by step through it. We also have an example data set where you can play around, where we provide you MRI data. You can then play around. We also provide some motor evoked potentials and get the approach running on your computer and test around. So, and of course apply it to new subjects in your lab. So, and it really describes in detail how to install everything and how to acquire the MRI. So every detail, which is usually short in a research paper, where the methods part is very compressed. This is basically a very detailed step-by-step description how to do it, and we also provide the code we used and we developed, and you can then readily run it, and how to prepare the participants and so on, how to set up the neural navigation. So especially for people who are new to TMS, I think it's a very nice description on how to do TMS mapping. So, okay, so let's go into the method itself. So in the first step, we have to acquire an MRI. So we provide the MRI parameters also, which are preferable to get a good image quality. In general, we only need a T1, but T1 and T2 makes the head models more accurate, because we can distinguish between cerebrospinal fluid and bone a bit better when we have the additional T2 image. And if you want to use anisotropic conductivity information in the brain, you can also collect a diffusion weighted data set. So usually the whole protocol takes about 30 minutes to measure all 3 data sets, T1, T2, and DWI. And yes, so if you have the MRI data, you can create the head model, and we also provide the scripts which are basically using SimNips, the software package for our electric field simulations we are using. And this also gives you the opportunity to refine the mesh in the region of interest. So, and here you see basically the script names we provide in the protocol, which you can run. So everything is based on Python, and we're just basically calling and handling SimNips. And yes, it's a very easy, basically a way to get your head models. So, in the next step, we can immediately come to the data acquisition. So, and now we have a very simple protocol, and it's actually way simpler than the traditional grid-based mapping. So, in our approach, we do not use a grid-based approach. We are stimulating basically with random coil orientations and random coil placement over the motor cortex. So the more chaotic it is, the better, which also makes the whole protocol very easy to apply. So you do not have to really take care that the coil is actually correctly placed or something. So you can really just sample single data points and then move the coil slightly, and then there comes the next TMS pulse, and you move the coil again slightly, and then there comes the next pulse. So you can basically not do anything wrong here. So and the more diverse you stimulate, the better it is. And here you see basically the arrows indicate now the coil direction and the coil placement here. And for every single coil position, we record the motor evoked potential. So, for example, here in our example data set, we have about 1,000 coil positions. Just to play around a little bit with, and the pulses were sent every 5 seconds. And for every of these coil positions, and there we have the motor evoked potential here and the extracted MEP amplitude. So after you conducted the experiment and you collected the data, we have to merge this experimental data, which means that we are using the recorded coil positions from the Neuro navigation, and we are assigning and merging each with the corresponding motor evoked potential. So we are just getting the pairs here right, and this is what this merge script is doing. Basically, it combines the data from the 2 systems, from the MEP measurement system and the Neuro navigation. So, and that's it basically from the experimental part. It is worth to mention that you see here, you can also measure or map multiple muscles at the same time. So then you just have more channels, and you have for every coil position, then you have, let's say here we are measuring 3 finger muscles at the same time, and so you can map multiple muscles without any additional experimental effort. You just have to put the electrodes in before you start the experiment. And in the post-processing, we will disentangle the individual fingers from each other again. So the next step now involves our electric field calculation. So once we have all the data and we have all the coil positions we applied in the lab, we have for every coil position one electric field distribution. For example, here at this particular coil position, we have this MEP and this electric field distribution. And for the next coil position, we have another MEP, which is, let's say, higher in this particular case, and we have another electric field distribution. So for every of these coil positions, we need to calculate the electric field, and we have a script for this doing it. And yeah, this is basically a step which takes some time, but we are also working on, of course, a bit faster solvers that it doesn't take so long, and it's running overnight or maybe in a few hours, let's say. It depends also a little bit on your hardware you are using. In this step, you can also, of course, use your own electric field solver. So if you do not want to use SimLabs, you want to use something else, you can, of course, do it. So as long as you calculate it correctly, it's fine. So this is your complete freedom. So okay, so now let's assume we have the electric field in every coil position. Now it comes to the actual mapping. So our approach is now based on nonlinear regression. So in our region of interest now here, the motor cortex and some parts around, we can now define our region of interest elements here. And in every element we have the electric field of all the different stimulations. For example, 1000 stimulation, we have 1000 electric fields for every region of interest element. So, and then we are going to use the motor evoked potentials here on the y-axis, and we are performing a nonlinear regression in every element. Now our hypothesis is that in the element of interest, so where we are assuming that the corresponding neurons, which are responsible for the finger twitch, are sitting, we should see a very nice relationship between the electric field we apply and the output we observe. So the electric field is now on the x-axis, and the higher the electric field is, of course, the higher the motor evoked potential should be. So this is our hypothesis here in this step. So, and you can see here in the left part, for example, it works very well. So this is a hotspot, and what we are going to calculate here is the R2 score, so the goodness of fit. And then for every region of interest element, we get one number for the R2 score, how well it fitted, and this is what we are going to plot here. So this is the R2 score over the different voxels. And for example, in an element where it's not really fitting very well, we get a low R2 score, and then the fit, for example, looks like this. So there is no clear relationship between the electric field and the motor evoked potential. So it is chaotic, basically. So these positions we do not assume to be responsible for our stimulation. So this is a very simple approach, but yeah, working quite well. And this is basically what this script 7 here is doing, and it's also not taking so long. Okay, so once we calculated the cortical hotspot, our mapping is basically done. So now we can say, okay, the R2 hotspot corresponds to our population of neurons we were looking for, which is responsible for this effect. And if you want to map, if you were mapping multiple muscles, for example, then you have to do the nonlinear mapping with the different MEPs only. So the electric fields you have already calculated, you just exchange this MEP variable here. You have to do the nonlinear regression again, and then you get a different hotspot where this muscle was fitting best, basically. And this is why you can map as many muscles you want, basically, in this approach. Just in the post-processing you have to replace the MEPs then and run it multiple times in the post-processing. So it's not any additional experimental effort. So once you, let's say, know the location of the neurons you were looking for, we can also do some optimization here. So let's say, okay, the black dot now is the hotspot of our R2 map, so of our goodness of fit, and now this is our target. We want to target. Now we want to look for a coil position, or for coil position and orientation, which most effectively stimulates this particular target. And for this, we do a very simple approach. It's basically not a real optimization. It's a brute force approach that we are trying a lot of different coil positions here. Here you see in the zoomed in value here. We are just trying to, or we are calculating the electric field for each of these coil positions in the brain. We are reading out the value at our target, and then we are selecting this coil position, which generates just the highest electric field in the target. And this is our coil position optimization. So we did this for some subjects, and then we went to the lab again and tested if this coil position we just determined was really the most effective one. So what does it mean now to be effective? It now means basically which results in the lowest motor threshold. So the lowest intensity what you need in order to stimulate the neurons. So if you slightly move the coil, for example, then you get a higher motor threshold. So now let's come to the mapping results. First, again, for different muscles, here you see we did it for different finger muscles, for the APP, FDI, and ADM, for example. The hotspots are super close together, which we also expected, of course, because the individual finger representations are expected to be very closely related, but we could see differences in the locations. And yes, here you see they are how they are located, and it's also interesting how they are oriented in the brain. Okay, now let's come to the validation experiment I was just talking about. So we're calculating the optimal coil position for each subject. For example, it was the central one here was the optimal one, and then we rotated the coil to a suboptimal position or orientation, and then we were measuring the resting motor threshold again. And if it's higher, then this is basically just showing that this position is not optimal. And then we were moving the coil a little bit around, and we're looking if by chance any other coil position resulted in a lower value, but this was indeed not the case. So over a huge group of subjects, and here you see basically the optimal or the relative motor threshold. In the optimal one, so the first dot here on the very left side was the resting motor threshold in the optimal one here, and then the other ones are for the other coil positions. In general, it was working very, very well. We had some outliers that, for example, here in this subject, there was another coil position was a bit more effective, but this was merely the case. So only the green subject here, and you can only tune the stimulator in percent, so from 1 to 100% output, and this is maybe 1% stimulator output. It was less, so it was super close. So this shows that basically the general approach of the mapping and of determining the optimal coil position for TMS works quite well. And this is again an overview about the whole approach. So we were starting from head model generation, then we went to the lab, randomly mapping TMS, we are calculating the electric fields. And with the MEPs and the electric fields, we were able to perform this nonlinear regression. And finally, we can determine the optimal coil position. So here we have the corresponding repository where you can download all example data sets and code from, if you are interested in to try it also in your lab. And now we come to the next question. If you are now, could I convince you to try the new mapping approach I just presented in your own lab or clinic in the future? Ah, nice. So it was.
So it has been convincing, Konstantin.
It was. This is good. Yes. Yeah, we were also surprised that it is working really well. Basically, and this was also our intention why we were writing this protocol, because a research paper alone basically is a nice to have and people are reading it, but it took us also some time to get everything running, to get all the electric field simulations running properly, to get all the nasty details basically to work. And this is why we published this protocol, which I think is a very nice way to really see how we did it and to get insights into it and to make it also used in other labs. So this was also our goal, that people could use it in the future. So now the question is, how can we do better? So of course we have a nice approach already, but we were still simplifying some things. Yes, we are making use of electric field calculations, and this is nice, but the brain is a complicated thing. So what we currently completely neglected is directional sensitivity of neurons. And we saw in other publications that the direction of the electric field can play a major role in how neurons are stimulated. And so far, we were only using the electric field magnitude for our regression. And the question now is, could we can we improve our mapping approach if we include information about the electric field direction in the brain relative to the cortex? And this we were investigating in detail in our most recent publication, where we were assuming different neurons in different cortical layers, and we wanted or we calculated an average response model, so an average threshold model, how the neurons respond when the electric field is applied in different angles to the neuron. So, and additionally, we also have this electric field, let's say, difference parameter, this delta E. So because a neuron is small, but it's not so small, so the electric field can change from, let's say, top of layer 2, 3 down to layer 5 or layer 6. So the electric field can change in its strength. So when you concern 1 or 2 millimeters of neuron height, which is often observed. So our most recent publication describing is published in Imaging Neuroscience. We're very happy to support also this new journal, this new open access journal, and I want to give you some insights what we found there and how we are going, or how we can use basically this direction sensitivity now also in our mapping approach. So we were looking at different cells here. So we were looking at layer 2, 3 pyramidal cells. In total, we had 24 of them. Here, just some examples. We have layer 4 cells, and we have layer 4 different types of layer 4 cells: small, nested, and large basket cells, which are inhibitory interneurons. And we have these layer 5 pyramidal cells, which are basically projecting down the spinal cord to the lower alpha motor neurons. And we were looking at the directional sensitivity of all of them. And our goal was to apply or to develop an average response model. So usually you do not want to play around or use all the detailed neural simulations in your simulations would take way too long. So our goal was to make a very easy to use model. And yeah, these are reconstructions from the Blue Brain project, and we collaborated with the group there, and they nicely provided us with the necessary data. And yes, these red neurons were scaled to human size according to the publications and the approach of Ibarra. And we also had to apply some reparameterization like myelination and ion channel properties, but this is very in detail described here in the two Ibarra publications. And so what we found is if we average now basically over all neurons in one population, and here you see the results for layer 5 pyramidal cells, just as an example. We have here, this color plot shows now the threshold, the electric field threshold until the neuron fires. So we are really looking at Hodgkin-Huxley equations here. What we are looking at, we were increasing the electric field until it was generating an action potential. And then we rotated the field again a little bit, and then we were calculating the threshold again. So on the x-axis here, you see this incident electric field angle here with respect to the soma-to-dendritic axis. And on the y-axis here, you see this is the parameter of the electric field differences between top and bottom in relative, so percent per millimeter electric field drop or increase. So sometimes the electric field can also increase. It depends on the location in the brain, so where you're looking at. So these are now averaged over all the neurons we had in this particular layer 5 population. And if we take now a crosscut here along this delta E is 0, so and now we just plot the directional sensitivity here, we see that the thresholds do increase for tangential electric fields and to decrease again when the fields are normal, so along the soma-to-dendritic axis. And this is exactly what we also expected. So if the electric field is perpendicular to the cortex, it is very effective. But in the past, this model was called the cortical Cullen cosine model. So you project your E field vector onto the normal component of your mid layer or cortex, and but this has a big disadvantage, because then with the cortical Cullen cosine model, you would assume that the tangential electric field would have an infinite threshold, so it would not be possible to stimulate any neurons with a tangential electric field. But this is basically not the case in reality. So this is a large oversimplification of the whole thing. And here you see in a dashed black line what the cortical Cullen cosine model would result in. So it would have an infinite high threshold if the electric field would be tangential and the projection would be basically 0. And here in this, you see the actual model recalculated, and this dependency is way weaker. So, and as the dashed red line now shows the magnitude model. So we applied previously, and the magnitude model basically does not assume any directional sensitivity, so that doesn't care which direction you apply. And when you look at this plot, you see that the magnitude model is actually not bad. So it's actually better than the cortical Cullen cosine model. And this was also kind of surprising to us, but we also saw it in the lab that tangential electric fields can stimulate neurons because the arborization, when you look at the actual neurons, you always have axon segments which are not, so the axon is not pointing perfectly downwards. It has segments which are also pointing, let's say, it's into the horizontal direction, and these can be stimulated, or these are stimulated by horizontal electric fields or tangential electric fields. And but the majority, of course, is pointing along the somatogenetic axis, and this is why this theta 0 or theta 180 degree is resides in the lowest threshold. Okay, so equipped with this knowledge now and with this map, we can now think, how can we use this information? So now we have a directional sensitivity, and how can we use it in our mapping? And this is now how we did it. So you saw the workflow does not change really much. So we stimulate random, we calculate the electric field, but additionally to the magnitude, we also calculate the angle of the electric field in each region of interest voxel, and we calculate this electric field drop delta E. So in what we are now doing, I took this map just from our previous result, and I was rescaling it, so normalizing it to an arbitrary value here, for example, theta 0 degree, and here delta rel is also 0, and then we get values between about 90% and 120%, for example, as a weighting function. So and now I let's say defined a so-called effective electric field. So the effective electric field is our electric field, which is depending on location and of course the incident angle theta and the electric field drop. And I'm going to divide this by the sensitivity map. So what does S mean? This S is basically this colored plot now, our neural sensitivity. You see here on the left. So a small value means a value smaller than 1 means this is an efficient electric field. So you need a low threshold. So and values higher than 1 means the electric field is inefficient. So we are operating here in the red region. So you need higher, you have to increase the field intensity to reach the threshold. And by this, we hope basically to correct our input-output curve, or to correct our electric fields here in a way that the I/O curves fits better. So for example, we have to do this for every single electric field, but this is only a very simple division here to just divide this map by the sensitivity map here, and now we have the effective electric field map, and then we are performing the regression with the effective electric field instead of the raw electric field, I must say. So, and our hope was that by this mathematical operation now shifts all points. So every point is shifted differently because the theta is different in every for every coil angle, and the electric field drop will be different. So every of these points will be divided by a different value of S, which is basically an element-wise division of these two maps here. And we hope that we could explain more variance basically by it to shift the points into the right location. So how does the results look like? So on the left-hand side, you see the result for the standard regression. And on the right-hand side, you see the neuron-enhanced regression. So I termed it just neuron-enhanced because now we are using neuron information. And you can see that the basic hotspot did not change, and this is also a good thing because our approach was also previously working, obviously from the validation, but we could gain a little bit of resolution here. So here we saw, okay, we have these, when we look here from the side, that we have some reflection here in the R2 scores, and these scores we could remove by applying this neuron regression. So our point spread function is now a little bit sharper. So by and this was we're very happy with this. So all calculations and efforts seem to be worth it. So and then we were looking, of course, at different subjects, applying neuron regression, and you see not really much of a difference here. So the differences are small, but they are still significant. So if we look at the R2 max values, so the highest R2 scores in these by applying these two approaches, we see that we could explain more variance over all subjects in average, and this effect is significant. And we did not observe any strange effects for some subjects, so it was almost every time improving. So, and this gives us confidence that we could use this approach as a standard method in the future. So, because it's also super easy, you do not have to do anything in different from the experimental point of view. It's just numerical post-processing alternative. So, just to use a more accurate model here. So, and indeed, our hope that we could explain more variance, was the case. Maybe not as extreme as I show it here on the lower left, but at least the R2 scores are now a bit better than before. So now it comes to the final point, TMS electric field optimization. So now you may wonder, okay, I defined this effective electric field instead of the raw electric field. And now the question is that we can, or we could go again or look back into our electric field optimization to find the best coil position. So, and now the goal is not to find the coil position which generates the highest electric field magnitude, but it's in the highest effective electric field instead. So we have to modify our goal function by dividing basically the electric field also by this sensitivity MEPs. So this is basically the approach here. We will use the same approach, but the results are still we are still working on. And the biggest questions are now, which are subject of current research, are how do the new coil positions look like in comparison to the old optimized ones, and are the new coil positions more effective? And as you may remember from our previous validation, we had two subjects where at two coil positions it was 1% lower. So maybe we can also be even more accurate here, and we can get small corrections on these optimal coil positions here by using this neuron enhanced method, including directional sensitivity. So, and now my final question is, if all the method would be implemented in a neural navigation system, would you use it? Basically, you do not have to care about electric field calculation. You do not have to take care about merging experimental data. So basically, everything would be integrated into an easy to use neural navigation, and the neural navigation system provides you the map. There is the hotspot, and there you have to most effectively stimulate. Would you use it? So you do not have to install anything. The neural navigation is doing everything for you, basically.
All right, I close the poll and share the results. It seems we have a cool 5% group. Would be nice to talk to them during Q&A and share and hear their comments about it.
Yeah, I'm very happy that people would like to use it. It's also very interesting for us to collaborate with basic companies to translate this scientific knowledge we gathered so far into practice. And of course, we want to do it as easy as possible for the people not to care about any of the nasty things when it comes to these model generation and blah blah. So I know that this can be a very time-consuming thing, and everyone could save a lot of time and do it without stress if it would be implemented really in a neural navigation system. So, and
absolutely, that's very positive result. So
yeah. Okay, so this is basically the end of my presentation. So thank you very much for listening. I'm happy to answer any questions.
So thank you, Konstantin, for this brilliant presentation, sharing your valuable insights on how to make motor mapping with TMS as efficient and effective as possible. I'm sure it provides some moments of reflection in the next roadmap planning for neuronavigation systems in the future.